Hey everyone, how's it going? In this video, we're going to learn about our body's cannabinoid system, which is a natural system of our body. And we're going to learn about how various endocannabinoids and exogenous cannabinoids can activate this system. So, endocannabinoids are messenger molecules they activate the cannabinoid system, but the prefix endo signifies that they are endogenous, meaning that these endocannabinoids, these messenger molecules, are produced naturally inside the body. And there are also exogenous cannabinoids, which are not naturally produced inside the body. They originate from outside the body. And this can come from plants, for example, cannabis. That's what the cannabinoid system is named after. It's named after your plant cannabis. And exogenous cannabinoids can also be synthetic. So you can have cannabinoids that are present in different medications and chemicals that are designed to produce specific effects in the body by stimulating or interacting with your cannabinoid system. So let's first take a look at the cannabinoid system, learn about the cycle of endocannabinoids inside the body, and then take a look at some exogenous cannabinoids both in the plant form and in the synthetic form and see what they do in the body. Let's get into it. So a while ago, we made a video detailing the general cell signaling framework with this acronym PPRRT. So messenger molecules must first be produced, then they are sometimes packaged, but sometimes they're not. Then after they are released from the vesicle in which they're stored or from the cell in which they're produced, they go off and produce responses, usually by binding to receptors, and these responses must eventually get terminated. So if you haven't seen this video, I recommend taking a look to get some background knowledge. I'll leave a link down in the description below. And endocannabinoids, which is the messenger molecule we're going to focus on in this video, they follow this same framework. So they are synthesized in the body, they are actually not packaged, and they get released, produce responses, and these responses must be terminated. So the main reason why endocannabinoids are not packaged is because they are lipophilic, and this means that it's very hard to package them. So usually messenger molecules that are packaged, like histamine, they are hydrophilic. Hydrophilic means that it likes water and it does not like fat. So what that means is if you have your histamine inside a vesicle which has a lipid membrane, because this histamine is polar and hydrophilic, meaning it likes water but doesn't like fats, it cannot escape through your lipid membrane because it doesn't like lipids. It's not going to be able to pass through this lipid layer. It's blocking it from exiting. However, endocannabinoids, on the other hand, are lipophilic, meaning they're the opposite. They like fat and they don't like water. So if you have endocannabinoids inside your vesicle, because they like fat, they will easily be able to pass right through your lipid membrane and exit the vesicle. So this makes it very difficult to store or package lipophilic messenger molecules such as endocannabinoids. That's the main reason why they're not packaged. As soon as they are produced, they are released outside the cell. So if we look at the first step of our signaling framework, it is production. We need to produce our endocannabinoids. Let's take a look at how this happens. So our bodies have two main endocannabinoids, and these are the ones we're going to focus on. So one very important fatty acid in our body is arachidonic acid. This fatty acid is involved in all sorts of different biological pathways and processes, and two derivatives of arachidonic acid are shown here and here in your blue boxes. So you have, I'm going to struggle to pronounce this, but phosphatidylphenolamine and phosphatidylinositol. These are two derivatives of arachidonic acid, and these are the precursors of your two main endocannabinoids. So these two derivatives of arachidonic acid can be converted and metabolized by various enzymes and go through these pathways in order to produce your two main endocannabinoids. So you have anandamide, which can be written as AEA for short, and 2-arachidonyl glycerol, which can be written as 2AG for short. These are your two main endocannabinoids. So now we know how our two main endocannabinoids are produced, but when are they produced? What triggers their production? The production of endocannabinoids is triggered as a regulatory mechanism to help control neurotransmitter activity. So let's take a look at this diagram here. We have a presynaptic neuron, a synapse, which is a gap between your two neurons, and then you have your postsynaptic neuron. So in our past video regarding the release mechanisms of messenger molecules, we've covered the process of how neurotransmitters get released from neurons and go and activate other neurons. So be sure to check out this video. I've also left this in the description below. But basically, in your presynaptic neuron are these vesicles containing neurotransmitters. When calcium ions enter via these channels, this triggers the release of your neurotransmitters out of your presynaptic neuron and into your synapse. And once it's in the synapse, these neurotransmitters can go and bind onto your receptors of your postsynaptic neuron, producing responses. So some of the responses that can take place when your neurotransmitters bind to the receptors of your postsynaptic neuron are, for example, calcium ions can enter your postsynaptic neuron, kind of like how here they entered your presynaptic neuron, except here your calcium ions entering your presynaptic neuron triggered the release of your neurotransmitters, whereas for your postsynaptic neurons, after your neurotransmitters bind to receptors, the response is an influx of calcium ions. Other possible responses are stress responses. However, these responses need to be regulated and kept under control to keep your body safe. We need some sort of regulatory mechanism, and that is where the endocannabinoid system comes in. So when calcium enters the postsynaptic neuron or other stress responses happen in your postsynaptic neuron, this triggers the production of endocannabinoids inside your postsynaptic neuron. Okay, so now we know how endocannabinoids are produced and what triggers the production of our endocannabinoids AEA and 2-AG in our postsynaptic neuron. So that covers the first step of the signaling pathway. Production is good. And now remember, we said that since our endocannabinoids are lipophilic, they are not packaged. So we can go right over to the next step of release. So because they are not packaged, as soon as anandamide or 2-arachidonyl glycerol are produced, they are immediately released from your postsynaptic neuron. Okay, so once some type of response like stress responses or the entry of calcium triggers the production of your endocannabinoids in your postsynaptic receptor, remember they don't get packaged, so right as they get produced, they can pass through the cell membrane and they can exit your postsynaptic neuron, and then they can make its way into the synapse. So you may notice that this looks a little bit different than other messenger molecules we've seen in the past, like histamine or acetylcholine. Those messenger molecules were produced in the presynaptic neuron, and then they were released, and then they bound onto the postsynaptic neuron. Kind of like the neurotransmitters that you find here. They're going from presynaptic to postsynaptic. But endocannabinoids, they work the opposite way. They're produced in the postsynaptic receptor, postsynaptic neuron, and then they're released and work backwards to the presynaptic neuron. So this is a unique type of cell signaling called retrograde signaling. Alright, so that covers the next step of release. Now let's see how these endocannabinoids produce responses. Now your body has two main types of cannabinoid receptors, which are CB1 receptors and CB2 receptors, which stand for cannabinoid receptor 1 and cannabinoid receptor 2. Let's first take a look at our CB1 receptors. Okay, so calcium ions, they enter your postsynaptic neuron, which triggers the production of anandamide and 2-arachidonyl glycerol, which are your endocannabinoids. And then they are released into your synapse. Now on your presynaptic neuron are CB1 receptors, which are a type of GPCR. Now I really recommend watching my old videos on GPCRs and GI proteins and how they impact adenyl cyclase and CAMP production. Be sure to check out these videos, I'll link them on the description below, and they'll really help understand this next component. So your endocannabinoids, AEA and 2-AG, they exit out into your synapse, and they go up to your presynaptic neuron and they bind to your CB1 GPCRs. Now, your CB1 GPCRs, when they're activated by your endocannabinoids, they activate GI proteins. And GI proteins, they lower the activity of adenylyl cyclase. And adenylyl cyclase is an enzyme which produces your second messenger, CAMP, which is responsible for all sorts of responses. So by inhibiting adenylyl cyclase, they lower the levels of CAMP. And in doing so, it changes the physical structure and conformation of your calcium ion channels, which inhibits calcium ions from entering. So the amount of calcium that enters will be decreased. And in turn, this decreases the amount of neurotransmitters that are released. So less neurotransmitters are released, and less neurotransmitters are released means that less neurotransmitters are binding to your postsynaptic receptors, meaning that less response is now happening in your postsynaptic neurons. So the amount of calcium ions that are entering are decreased, and so response levels are decreased. So you can see it's kind of it's a negative feedback loop to keep response levels under control, because normally what happens is calcium ions enter, neurotransmitters are released, they go and bind onto receptors, producing responses such as calcium ions entering. And this is fine, but we need to keep these responses under control in order to prevent things from getting dangerous. So your endocannabinoids are produced, which go and they bind to cannabinoid 1 receptors. Cannabinoid 1 receptors on your presynaptic neuron, activates GI proteins, which inhibits adenylyl cyclase, lower CAMP, so less calcium is entering, less neurotransmitter release, less binding to receptors, less calcium ions entering, and less stress responses in your postsynaptic receptor. And via this negative feedback loop, the endocannabinoid system helps keep responses under control and helps keep response levels safe. CB1 receptors are mostly found in your central nervous system. CB2 receptors, they're also GPCRs. They also inhibit adenylyl cyclase and lower levels of CAMP. However, they're mostly found in your peripheral nervous system and immune cells. Their responses often involve regulating immune responses and keeping inflammation and immune responses under control. So they're both involved in regulation systems, controlling response levels and keeping them under control. But CB1 receptors, they focus on regulating neurotransmitter release. CB2 receptors, they focus on regulating inflammation and immune responses. Alright, that's another step of the cell signaling framework down. Now, let's look at how these responses are terminated. So your two main endocannabinoids, which are anandamide and 2-arachidonyl glycerol, they are terminated by being reuptaken into neurons and degraded by enzymes. So 2-arachidonyl glycerol is taken into the presynaptic cell, and it is degraded by an enzyme called MAGL, which stands for monoacylglycerol lipase. So MAGL, it breaks down 2AG into arachidonic acid and glycerol. On the other hand, your AEA, your anandamide, it gets uptaken into your postsynaptic cell instead of your presynaptic cell, and it gets broken down by an enzyme called FAAH, which stands for fatty acid amide hydrolase. So FAAH, it breaks down anandamide into ethanolamine and arachidonic acid. So by breaking down your endocannabinoids, the body ensures that your cannabinoid responses don't go out of control and inhibit neurotransmitter release or immune responses too much, because neurotransmitter release and immune responses are very important. Everything in the body must remain in a fine balance for proper functioning. Your cannabinoid system is necessary to keep responses like calcium influx and stress responses under control, and your termination mechanisms are necessary to keep your endocannabinoids under control, and that covers the last step of termination. Okay, so now that we understand our cannabinoid system and some of our most common endocannabinoids, let's now talk about exogenous cannabinoids. So remember, exogenous cannabinoids are exogenous, meaning they are things that come from outside the body instead of being produced naturally inside the body. and they enter the body and they interact with our body's cannabinoid system to produce different responses. Now, exogenous cannabinoids, they can include, they can be synthetic, meaning man-made, produced in a factory or a lab, and this is usually found in different medicines or chemicals designed to produce specific responses in the body. And the other main source of exogenous cannabinoids would be the cannabis plant. This is what the cannabinoid system is named after, cannabis. So let's first talk about the cannabis plants. The cannabis plant has many chemicals called phytocannabinoids. Phytocannabinoids are chemicals in cannabis that interact with the endocannabinoid system. And the two main phytocannabinoids chemicals are delta-9 THC and CBD, which is short for cannabidiol. So let's take a look at what these two main phytocannabinoids do. Delta-9 THC, it mimics the endocannabinoid we talked about called AEA. It mimics AEA binding to CB1 and CB2 receptors, and it acts as an agonist. It stimulates these receptors to produce responses. Now, delta-9 THC, it causes psychoactive effects. So this is the compound in cannabis that causes you to become high when smoking weed or eating edibles. The second main phytocannabinoid, CBD or cannabidiol, it has a lower affinity to CB1 and CB2 receptors, and on its own it's actually not psychoactive. So it doesn't really act as much as a agonist, but rather it acts as an allosteric modulator of your CB1 receptor. So on its own, CBD doesn't really do much, but when using CBD in combination with delta-9 THC, it can alter the effects that delta-9 THC has. It can alter its psychoactive effects and responses. And this is the difference between a good high versus a bad high. So using delta-9 THC alone can cause psychoactive effects and cause you to become high, but using delta-9 THC in combination with cannabidiol can alter these effects and smoothen them out, and oftentimes can create a more euphoric and pleasant high experience. Another another thing that CBD does is it inhibits FAAH. Remember, FAAH is the enzyme that degrades AEA. So by inhibiting FAAH, this allows for increased levels of anandamide or AEA. Other than CBD and delta-9 THC, there are also other phytocannabinoids present in cannabis that contribute to other things such as the flavor of weed and the smell of weed. So that covers the cannabis plants. Now let's talk about synthetic exogenous cannabinoids. So as we said, synthetic exogenous cannabinoids found in medicines and chemicals can also interact with the body's endocannabinoid system to produce various effects and responses. So Epidiolex is a prescription cannabinoid medication to help treat epilepsy. Nabilone, it mimics THC for sleep and pain relief. Nabiximols, it has cannabis extract and it helps relieve pain and cure multiple sclerosis. And acetaminophen, which is found in Tylenol, it acts as a CB1 and CB2 receptor agonist, and it also inhibits the uptake and termination of anandamide or AEA, increasing anandamide levels. So, in conclusion, the body's cannabinoid system is essential to keeping responses like calcium influx levels and stress responses under control. So, the responses like calcium entering your postsynaptic neuron or other stress responses, they trigger the production of anandamide and 2-arachidonyl glycerol, which are your main endocannabinoids. Now these are released from your postsynaptic neuron, and they can go and bind to your CB1 receptor, which is mainly a part of your central nervous system, or CB2 receptors, which are mainly part of your peripheral nervous system and are found on immune cells as well. Both CB1 and CB2 receptors are a type of GPCR. CB1 receptors are linked to GI proteins, which consequently lowers the activity of adenyl cyclase, which consequently lowers the production of your second messenger, CAMP. And this changes because basically you have calcium ion channels for calcium to enter, but the reduction in CAMP changes the conformation of these ion channels to make it more difficult for calcium to enter, and that prevents the release of neurotransmitters. So you have less neurotransmitters being released, less binding onto receptors, and therefore you have a decrease in calcium influx and a decrease in stress responses. So it's an effective way of keeping these responses under control. Now, in order to keep the cannabinoid responses under control, we need to be able to terminate the responses of anandamide and 2-AG. So what happens is your AEA and your 2-AG, from your synapse, your AEA is uptaken into your postsynaptic neuron, where it gets degraded by your enzyme FAAH. On the other hand, your cannabinoid 2-AG, let me do it in a different color, it's getting messy. 2-AG, it gets uptaken into your presynaptic neuron, where it gets degraded by your enzyme MAGL. So that's how your cannabinoids are terminated. And then you got exogenous cannabinoids. So cannabis plants, they contain phytocannabinoids, which are chemicals in cannabis that interact with the cannabinoid system, and some are very simple, mostly contributing to the flavor or the smell of weed. But the two main ones that interact with the cannabinoid system are delta-9 THC and CBD, which stands for cannabidiol. So delta-9 THC, it activates CB1 and CB2 receptors, and it causes psychoactive effects. And CBD, on the other hand, is an allosteric modulator that changes and refines delta-9 THC's normal effects. It also inhibits FAAH. And that concludes this episode. I hope you were all able to get a good understanding of how the endocannabinoid system works, why the system is so important, and how chemicals in cannabis and medicines can interact with this system. Thank you guys so much for watching. I really appreciate all the support. If you liked the video, please subscribe and like. It would mean a lot to me. And if there's anything you'd like me to make a video on specifically, or if you have any questions or feedback, feel free to leave a comment below. In the next few episodes, we're going to look at some more messenger molecule pathways, maybe nitric oxide and steroids, prostaglandins. We'll see. But until then, thank you again for watching. Have a great day, and I'll see you guys next time.